Hi guys, today we'll be talking about clinical drug development and its rationale. I'm Sheba David. You have been seeing me for quite a number of lectures. Let's start with this. We're talking about drug discovery and preclinical testing. First, let's talk about drug discovery. What actually is drug discovery? If you look at this, first we have drug discovery and then we have the preclinical development, then we have the clinical drug development. Look at all this stuff coming over, and when you come over into the clinical drug development, we see that there's approval. So which means that in this approval, we have somebody who is very important to be playing a big role. Who is that? FDA. So what's FDA? Food and Drug Administration. This is the consumer watchdog for numerous products. You know this, drugs and biologicals. This is the food, medical devices. You all know this definitely. And look at this with my name on it as well. What's that? The animal feed and the drugs as well. And finally the cosmetics, all the things that we love. So what is this important about? IND and NDA. These two are the jargon which is very important for FDA. So what's IND? Investigational New Drug. What's NDA? New Drug Application. What does this have to do with all these processes? Look at this. Preclinical, clinical. All these things has to be done here. You need to submit the IND here and then you need to submit the NDA. And remember, FDA is the important watchdog for all these processes. Now, what's NDD, the new drug development? It's all about a highly regulated process, again by the FDA, and this drug development process is about research and development in the lab, testing in the animal models, and several phases of testing in the humans. First, we'll talk about R&D. What do we have in R&D? Target selection. What does this mean? Target selection is about taking a drug and then you want to see whether this drug is appropriate or not. And then we find about lead finding. Is this a lead appropriate for this particular disease? And then if it is applicable, let's say that it's about 60%, then we want to optimize it to about 80 to 19%. So that is about lead optimization. And then about the pharmacological profiling. What is this? We want to see whether it's applicable pharmacologically. That's about R&D. So how do we do it preclinical testing? We take a drug. Is it possible that we take a drug and then give it to a human being? We can do that. So we need to do in preclinical testing. Look at those guinea pigs, lovely ones, aren't they? They are the ones who are sacrificing themselves for the greater good of the society. What do we have here? Testing in animal models. When new drugs show promise in the lab testing, they are tested in the animals, and that's what is called as a preclinical studies or preclinical testing. And then what do we do in preclinical testing? We test toxic and pharmacological effects, genetic toxicity, ADME, the jargon of pharmacology, and all the others. Again, remember, FDA requires this. What is FDA? Food and Drug Administration again. So this is all about preclinical testing. Then we talk about in vivo experiments. So what is vivo? Vivo means living things. So here in living things, when we talk about it, it's about taking animals, the rats or the guinea pigs. Once you take them, then you test for a drug. It can be for any disease. So it can be for hypertension in a rat, or it can be for diabetes, or name it, any disease that can be done. What is the advantage? It's closely approximate the situation in a man. And we can monitor the drug effects in one experiment itself. And we can determine where does this drug go, to this organ or to the lung or to the kidney. We do not know, so we can test out in the animal experiments. What are the disadvantages? Of course, it's crude, and of course the mechanism of action is based mostly on the circumstantial evidence. And of course, animals are to be sacrificed. In vitro experiments. So what is vitro? Vitro means glasses. So we do them in the glass beakers or gas cubicles or whatever it is. What is the advantage? All the disadvantages in vivo experiments can be nullified. Several different tissues can be used. For example, we can use heart separately, we can use kidney separately, and we can put a drug and see whether this drug is being acting on that particular organ and not on the whole body. And what's more important, it's easier to control or modify the experimental conditions because we are not using the whole animal at all, so it's very easy. What's the disadvantage? The environment is not exactly the same. It's just one organ, so we can't estimate the whole thing accordingly. And drugs which change or metabolize and which are pro-drugs, we can never find out what was the effect of it. So, to conclude the whole thing, we have drug discovery, and that would take about 2 to 5 years, preclinical development and all these stuff, then we go into the clinical development. Today we saw about drug discovery and preclinical treatments, and here we were talking about the animals sacrificing themselves for the greater good. Thank you so much for listening to this, and let me see you all in the class for the flipped classroom. Thank you once again and enjoy your learning.